On Tuesday, 11th of February, we are bringing back one of the hottest topics for our online industry meetings. I'm talking about biosensing. Last year, the online industry meeting on biosensing brought together many of our members showcasing what they can do to measure biomarkers using integrated photonics and laser technology. This year, we want to have an end-user driven discussion. We want to match the needs of those end users with the technology of our corporate members. That's why we have invited companies such as Pacific Bioscience, MagBiosense, Quanterix, and the engineering facilities of TNO in biosensing. We're gonna match this with the technology of many of our members, to mention a few: Bioherent, Surfix, Airware, LIGENTEC, Leuermann, Fraunhofer HHI. Let me highlight that this meeting is only possible because of the support of some of our corporate members. I would like to thank our sponsors, OpenLight, OptiWave, LIGENTEC, and Fix. Do not miss the opportunity to voice your opinion and pitch the technology of some of the end users in the biosensing market. See you on Tuesday, 11th of February at 10 a.m. in Washington or at 4 p.m. in Europe. See you soon. Ladies and gentlemen, it is time. We've been working very hard for many months until we finally are here in front of you to discuss our favorite topic. We are only a few weeks away from OFC. We just came back from Photonics West. We are excited about new developments, but the whole idea is that we can use those developments to improve the quality of life, to live longer and healthier. And for that, we need to measure some key biomarkers, and that's what we are here to do. The whole meeting today, the next 1.5 hours, is to make sure we connect all of you with a few end users who are here trying to find partners, suppliers, customers, investors, and talent, and all that is in front of them. Let me start by saying that this meeting is possible because of the hard work of our technical directors, and especially Dr. Helena Diaz, who has been discussing with many of you to make sure that all of you can provide technology and can provide support to this fantastic lineup. Let me start also by sharing with you a couple of ideas. The first one is that this is not the standard webinar. If you came here to just listen, maybe you can switch off the laptop and continue working. This is not a webinar. This is an online meeting. So all of you have a purpose here. I would like to give you a task. Think about your answer for the Optica question. What can you do for the others in the Zoom room, and what can the others do for you? And this is the rules of the game. We have a few integrators or end users, namely Pacific Bioscience, MagBiosense, TNO, and Quanterix, who are here looking for partners, and we have a few enablers. These are just an example. All of you are here as enablers. Bioherent, the biosensors Bioherent all the way from Malaga, Airware, Surfix, LIGENTEC, Leuermann for the photonic integrated circuits and activation surfaces, and Fraunhofer HHI are just examples, but all of you are here, and if you have any questions, comments, you want to share something with the room, the microphone and the spotlight will be coming to you. I would like to highlight that this is now the agenda for all online industry meetings from here until 10th of February 2026, a whole year in front of you of online industry meetings. If you want to show your technology at any of the upcoming online industry meetings, all you have to do is to send us an email and say, hey, I want to present at the fiber sensing meeting or at the gas and liquid sensing meeting. I want to make sure that the lighting meeting on the 6th of January has my technology on it. Make sure that you tell us, because of course, the meetings have limited room. The sooner you provide us with the information of your interest, the sooner we can make you happy, giving you the spotlight at the right industry meeting. Also, let me say as well that we are doing this because we support our corporate members. If you want us to continue doing our job, and we want to continue doing our job, join as a corporate member. Half of you are corporate members, the other half are potential members. If you want to know some of the membership benefits, Here they are. We provide access to network, market reports, great events. I would say the best events in the world, great discounts if you want to exhibit or you want to buy any of the Optica products, lots of promotion, and of course, lots of engagement. Please join us, because if you don't join us, then we can send you our CVs and join you. Please join as a corporate member. I also would like to also say that we have now the largest industry library of any association. If you're looking for market reports and you're a corporate member, or you're looking for any presentation ever given at an Optica industry meeting, you can access them by going to our online industry library. All corporate members have full access to every single report in that library. Please make use of it, because we spend a lot of money and a lot of effort making the most complete industry library. And also, let me just conclude by saying that this meeting is in Zoom, but also live broadcasted in YouTube. So let me say hello to the YouTubers of the world. Hello, YouTubers of the world. If you have any question or comment during the meeting, all you have to do is write in the chat and I will read in the room. And of course, this is also valid for the people here with me in the Zoom room. If you have any question, comment, challenge, write so in the chat. This is a live meeting in which we all want to participate, all want to discuss. And to start, and with no further ado,
I gave him the flow with numbers on Friday, and I need it by Thursday.
So you are mute.
Thank you very much, Helena. And let me start by saying that this meeting is now focused on end users. Last year, we have another biosensing meeting in which we had all the technologies that we can use for measuring biomarkers. This time, we wanted to have an end user-driven discussion. And to start, we couldn't start any better, right, Helena? We went to the market leader on biosensing worldwide. We went to Pacific Biosciences. Ravi, thank you very much for being with us today. Tell us what you can do for others and what others can do for you. The floor and the attention of everyone here online, we are global today, goes to you, Ravi.
Thank you, Jose, and thank you, the Optica staff, for inviting us to share what we are doing at Pacific Biosciences. It's a pleasure to speak to all of you. I'm going to tell you a little bit about how we at PacBio are leveraging optical technology or advances in optical technology to do single molecule detection and really to leverage that into DNA sequencing. So most of you know us already, but for the new folks, we are an end-to-end service provider for sequencing. So we make the entire system, the instruments, the consumables to do sequencing, and then the software and bioinformatics tools to interpret the sequencing data. We've been around since 2004. We went public in 2010, and we've sold about 1,300 sequencers worldwide. We're pretty much known as the gold standard in terms of high accuracy DNA sequencing. And this whole technology is really built upon my colleagues, both past and present, who have really contributed to some very advanced R&D to create this technology and to bring it to market. So I'll tell you a little bit about how we actually do sequencing and what is the heart of the sequencing technology. So what we do is we first of all immobilize DNA polymerase, which is the natural enzyme that sequences your DNA or synthesizes your DNA inside all our bodies. We modify that enzyme to slow it down, but we then immobilize that enzyme and DNA strand at the bottom of an aluminum film. It's basically a zero-mode waveguide that allows us to create a viewable window, a very small viewable window, and separates that reaction site from the rest of the background. The technology is fluorescence-based detection, so we have illumination light or excitation light at a certain wavelength, and then we are detecting labeled markers for each of the different bases, and each of those different labels basically allows us to distinguish, there are only 4 bases of the DNA, which is A, C, T, and G, so each of those markers allows us to distinguish those different bases. And the way the technology works is when you're sequencing and you're incorporating a single base, say you're incorporating an A, then you get a pulse of light in A, and you detect those photons. And then once those bases are incorporated, the label gets cleaved and floats away into the background, and that moves away from the viewable window. So that's how we are able to detect the different bases. The last trick to get super high accuracy in terms of DNA sequencing is we are able to, through chemistry and through some other tricks, go over a single strand of DNA multiple times. So by going over the same strand again and again, we take the error, which is essentially random error, out of the equation and thereby get accuracy, which is what we call consensus accuracy of sequencing, and get super high accuracy basically throughout the entire strand. So when we first released our product, we were essentially doing the structure on the left, which is a free space-based optical architecture, where we had essentially lasers going through an optical train. We had an objective that basically collected the signals from a passive chip, which provided the
I
confinement.
won't take any coffee anymore, but
and thereby collect the signal coming out of the chip and separate that into 4 different cameras. This, as you can imagine, was fairly bulky. Our first system was the PacBio RS, and it was pretty heavy and pretty large, and that provided us a challenge for scaling because, as you can imagine, the more number of zero-mode waveguides or the more number of reaction sites we have, the more number of DNA molecules we can sequence in parallel, and the more number of molecules we can sequence in parallel, the lower our cost is. So what we came up with was, well, how about we actually do a monolithic integration on top of the CMOS sensor itself of the various elements of the optical chain? And what I'm talking about is essentially integrating excitation through a waveguide-based illumination and the collection through integrated filters and lenses on top of the CMOS. Now, individually, these technologies had been done, but they had never been done together to create this kind of integrated optical architecture. And that's what we were able to do with our Sequel product line. And we have released now 3 platforms based upon that product line. And so how could we actually do it? So there were 2 fundamental parts that allowed us to achieve this. Number one was leveraging silicon photonics to create custom solutions. So we use a lot of photonic design tools. Here you see a numerical simulation on the left to create waveguides, different kinds of lenses. Here's the Fresnel lens there, different kinds of filters, and then different optical elements that allowed us to create the confinement very close to the waveguide. So we are able to immobilize the enzyme at the bottom of that confinement structure and then illuminate it with the waveguide and then collect the subsequent photons and direct them to individual pixels on the CMOS sensor itself. I would say, the biggest thing that actually helped us to do this was leveraging partners which had experience in silicon photonics, but also where both design and fabrication teams were working very closely together, because this is, as you can imagine, to do this in the visible spectra versus doing this in infrared, it poses certain challenges. And really, to make these processes high volume capable, we had the process engineers and the designers all working very closely together to fine tune the materials and the processes to come up with a manufacturing solution. The second big technology advance that has enabled us is really the advances in CMOS image sensors. And as most of you know, right, we have seen the whole trend of shrinking pixel sizes going from front side illumination to backside illumination to stacked. And we have been riding this wave of increasing sensitivity and increased throughput in terms of pixel density and better collection efficiency to detect less and less number of photons and to pack more and more reaction sites onto the pixel, onto the sensors. So as I mentioned earlier, the number of pixels on the sensor is directly proportional to the remote waveguides that we actually have on the sensor, which is directly proportional then to the throughput and thereby lowering our cost. I don't think we would have been able to continue the scaling had others before us not actually been able to create these advances in CMOS sensors. And we have now actually progressed all the way to the right here. We are looking at stack sensors to do our sensing. So what's the result? As you can see on the graph here, we've been able to greatly reduce our price per gigabase of sequence, which is essentially the metric for our customers, by going from a free space optical architecture, which was the RS system, to now the Revio-based architecture, where we are actively, doing integrated sequence on these sensors. Right now, we are about 2,500 genomes a year based on the Revio system, and we are not stopping yet because, as you can imagine, that the optical-based technology actually does not have a limit from the two slides that I showed you before. There are other scaling factors that we can get, and this is where I would basically look to this community and say, help us, with newer materials, newer designs, bigger fabs going to 300 millimeter, and trying to put more and more integrated functions onto the chip. Right now, we have a coupler on the chip. We are thinking of more modulators on the chip, and so on and so forth. Okay, I think that was my last slide. So hopefully you all got a flavor of what we do at Pacific Biosciences and how we are able to leverage advances in optical technology to do that. I'd like to then acknowledge both all my team members, past and present, for this work, and also the Optica staff, Jose, Elena, and the rest of the staff, for inviting me to share this work with you.
Thank you so much. Thank you so much, Ravi. This is really nice. So, I mean, all this technology. I mean, the architecture is what really makes PacBio unique. But one of the things that the people is always thinking in this kind of thing is like how does the PacBio's HiFi sequencing compare in accuracy and the read of the length to existing short read technologies?
Oh, that's a great question. So when it comes to the read length, the average read length that we are sequencing is of the order of 20,000 to 50,000 kilobases. That's kind of what we are talking about. The read length in the short read based technologies is somewhere between, depending upon the technology, 200 base pairs to 500 base pairs. I think the mode is at more around 200 to 300 base pairs. So what that allows us to do with the read length being that long is it allows us to go over long regions of the genome, and thereby, when you're assembling a puzzle with very big pieces, the full puzzle that you assemble is super accurate. Whereas when you have really little tiny pieces, you don't really know where those tiny pieces all fit in. You may come up with a picture, but the picture is going to be very granulated. It's going to have some errors in accuracy. So when it comes to the full genome, like the actual telomere to telomere genome, it's pretty been proven that the long read sequencing technology is the way to go. Whereas in marker detections, which are looking at very short segments of the DNA, yes, the optical technologies, because you're looking at counting signatures and you're looking at many of these short segments in parallel, then the short read technologies win. So I think there's two different technologies. They each have their own strengths and applications, and that's how the field is most likely going to progress in the future. It's a great question.
Yeah, thank you. So the other thing is like, what is the primary market opportunities for biosensing, and what are the challenges for these technologies that PacBio has?
So, what we really have at PacBio is a super sensitive microscope, right? It's a single molecule detection technology. Now, you can apply that technology to other kinds of biosensing detection as well. There's, no reason why you could not apply this to proteins or to other, not just DNA sequencing. So, the potential of the technology is a large. In terms of the market itself, the sequencing market is about a $6 billion market. It's supposed to grow, whatever you want to believe in market research reports, but it's supposed to grow at between 8% and 10% over the next couple of years to get to 8 billion in 27 or something like that. I believe so. So that gives you a relative size of the market. Now, similarly, there are other markets when you look at biosensing technologies. You know, I think we can. So I don't know whether I'm answering your entire question, but I think when you look at leveraging this technology in sequencing, there is a market, and then if you go into parallel TAMs or parallel markets, then there's the opportunity is much bigger.
Yeah, no, made totally sense. Yeah, and the other thing is like, I mean, most of the people here is very highly technical for biosensing, but there is something that for the people is, especially because this is going to be then broadcast on YouTube and it's going to see for all kind of people. When the people think in biosensing, think in medical technology, okay, but there are so many other applications. So what applications is PacBio focusing on?
Okay, so within the market of DNA sequencing, we serve all kinds of customers. Medical technologies or human genetics and oncology are two big segments within the market, but we are pretty much the market leaders when it comes to plant and animal genomics. So engineering crops, for example, when you think of like drought-resistant crops or disease-resistant crops, and understanding complex genomes of different species across the planet, that's a big market. And then there's an emerging field, as many of you guys know, about synthetic biology, CRISPR-Cas9, where you actually edit the DNA. We call those the emerging markets itself. So these are the different kinds of markets that we essentially play in. And most of our customers are, there's academic institutions, CROs, and then there is the biopharma companies that leverage us, and then of course as you mentioned, clinical markets is huge, and that's really where a lot of the growth is, and that's really where we can realize our mission of actually using these technologies to influence human health, and that's core to what we do at PacBio.
Yeah, excellent. So we have a question. Stay here. Sorry for a minute. Yeah. So Abraham Kipnis, are you around? Can you please open your microphone and ask yourself your question? Abraham, are you around?
Hi. Sorry. Yes.
Yeah.
Yeah. You mentioned in the future you were thinking about integrating modulators onto the chip. So I was wondering what would be the purpose of that, or the potential benefit you gain from doing that.
Yeah, so the whole idea, you give that as an example of a technology element that you can actually integrate. Like I mentioned, what our job is right now to bring, say, the illumination light, which is free space, all the way onto the chip in waveguide. So we translate that through a grading coupler and bring that laser light in. Now, as you can imagine, depending upon the coupling, depending upon the speed and the accuracy at which we want to do that, there's variation. And so having on-chip elements that allow us to minimize those variation and to do the coupling very effectively, those are examples of elements that we would want to actually put in.
Okay, thank you. So we also have in the room, and this is one of our enablers coming from Fraunhofer HHI, sorry. Okay, so Jakob Reck. So Jakob Reck, he works on photonics biosensors and integrated optical systems. And Jakob, if you please open your camera and your slide and share what you have to say on the work of Fraunhofer is doing on this.
Of course. Thanks a lot. And hi, everybody. So yeah, hi from Berlin. I follow up on Pacific Bioscience with integrated optics. So we at Fraunhofer HHI, we engage in a photonic platform for disposable multi-parameter point of care testing. that is label-free. So I think Fraunhofer HHI is known for decades for investing in the telecom community, but we're also doing much more in, for example, integrated sensing. So what you can see here is our prototype readout system, and especially on the corner in the left, the PICs we are producing and manufacturing in-house. And also we are doing microfluidics, wafer-based microfluidics. So the topics are here for us, medical, life science, hygiene, and also water testing for many different suitable biomarkers. So yeah, we have this in-house fab for silicon nitride, but also for indium phosphide or polymer materials from wavelength from visible to near infrared, and also doing much hybrid integration with these different material platforms, and also automatic assembly of, for example, lenses and other micro-optical elements. So and that not only for a small production, but also for medium-sized productions. So what we offer is a photonic system development from design to module, and with all the back-end and packaging capabilities. We are also looking for partners, for integrators for our technology, and also device developers and biofunctionalization specialists for our biosensors. And with this said, I think the 2 minutes are filled. So thanks.
Thank you so much. Thank you so much, Fraunhofer HHI, so corporate member. And my first question to you is going to be, how do you do the way of the alignment in this kind of systems?
Yeah, of course, it's a special system here. So we do it with tolerant grating couplers. So we have a coupling system based on grating couplers with a tolerance of around plus minus 25 micrometer. And that is something you can do with a passive aligning process. So you just have the PIC on a subcarrier, and then there's some kind of a magnetic system which takes the PIC in place and relative to the fiber array. And all the other things, all the lasers, all the photodetectors, and so on, the DHQ system are in the box beneath.
Okay, so and of course, I mean, the Optica question. What you can do for others, which you already say, although Fraunhofer can make so much more in biosensing, and what the industry here can do for you.
Yeah, of course. So we are experts in photonic integration of different materials. So what we offer is this development from a design perspective. So you come to us with an idea, and we do at first some kind of blue sky thinking, and then we go to the module design, and we can do all the manufacturing and from fab over hybrid integration here in-house in cooperation with us. And what we or what you can do for us is, as this is a very interdisciplinary topic, we are searching always for application partners. So we are not the device sellers. So we want to cooperate with the companies in the field. For example, with the medtech device developers.
Yeah. So this is going to be for both of you, for Ravi and for Jacob. So what are the scalability challenges but you have when you are in the roadmap for mass production?
Mm-hmm. So I think from a photonics point, I just can talk about the photonic perspectives maybe. I think our biggest challenge, as we have these multi-parameter tests, is the mass production of the surface functionalization. So we have these photonic chips. They have wafer-based. They can be made in mass production, but the biofunctionalization for these ones we see as a challenge in future.
Yeah, I think that challenge is not unique in terms of surface functionalization for biomolecules, because most of our technologies that we leverage are wafer-based technologies, which are fab-based, which CMOS compatible processes are not known for biocompatibility in some sense, right? So I completely agree with Jacob that is a challenge. I think the overall challenge for us is, and something that we spend a lot of time on, is to ensure that the yield of our devices, because we are monolithic, we are doing a monolithic integration on top of CMOS. So, at any step, you can essentially scrap the wafer. So what we really want to make sure is that we put a lot of energy on is to come up with processes which have super high yield at the end of the manufacturing. So that's a very important scalability challenge. And in order to do that, it's not just the fab's job, it's also the designer's job, right? So you want to make sure that the design is capable of the tolerance that the fab processes are able to come up with. And we continue to work on those challenges all the time.
Yeah, I think this is one of the things that is big for this industry, because I mean, there are so many changes now, architectures different, and everything, and going into mass production, and all the scalability is not straightforward. Okay, so our next speaker is coming all the way from Israel. So Amos Danielli is the founder of MagBiosense, and they specialize in point-of-care biosensors. So the floor at the attention is all of you, Amos. Amos, you there? We cannot hear you.
Sorry, I was muted.
Yeah, you are.
Yeah. Thank you again for the kind invitation to present to you our work at MagBiosense. First, let me say I'm the founder and CEO of MagBiosense, but I'm also the head of the Optical Imaging and Biosensing Laboratory at Bar-Ilan University in Israel, and the head of the bioengineering track in the university. So MagBiosense focuses on developing very sensitive detection tool for rapid and highly sensitive detection of biomarkers. And the way we do it is we use fluorescence markers. And the challenge with fluorescent markers is that biologists, they know how to specifically bind a target protein or an antibody or a specific DNA sequence with a fluorescent molecules. However, to see fluorescent molecules, you need to shine laser light onto the solution. But the challenge is that if you look at the size of the room that you are looking at, that you are sitting in, the size of the laser beam compared to the room you are sitting in is about the size of my fist. And therefore, at low concentration, you only have few fluorescent molecules inside your detection volume, and the signal is masked by background noise of unbound fluorescent molecules. What we do with the optical modulation biosensing is something that other people are doing as well. We tag the target molecule with a magnetic particle. This way, we create a sandwich where the target molecule is detected by both fluorescent molecules and magnetic beads. But our patent is to aggregate all the magnetic particles from the entire room into your fist, into the flashlight. And now, instead of 2 or 3 fluorescent molecules, you have thousands of fluorescent molecules, and the signal increases by orders of magnitude. Not only that, to separate the signal from the unbound fluorescent molecule from the signal on the beads, we modulate the laser beam. We look at the background and we look at the beads. And therefore, we can subtract the background noise from the signal from the beads that we look at. So to summarize, we have 2 advantages. First, we concentrate, so we can read a 96 well plate. Each well we can read within 5 seconds or 6 seconds. and aggregate them very rapidly. Here we go, every 5 seconds, we move to the other well. And the second thing is the fact that we modulate the light, so we can look at both the beads with the fluorescent signal and the background, and actually subtract the signal from the background from the signal to the beads. Therefore, we don't need to do washing or separation steps that are usually incorporated in heterogeneous assays such as ELISA. So MagBiosense built a device that is currently a laboratory device that can read the 96 well plate within 10 minutes. You can actually specify in each well what do you want to detect and what is the threshold that you look at. It's very quantitative because you have very high sensitivity to different amount of fluorescent molecule per bead. So you can detect very few fluorescent molecules in solution and up to millions of fluorescent molecules per bead in solution. And this is what we did. This is our technician fill in the device. You can press scan, and then it scans and it goes into operation mode where it can scan all the required wells. So overall, we have few advantages over current technologies that are utilized in laboratory settings, such as ELISA. ELISA has a turnover time of 4 hours, whereas our turnaround time is between 15 to 60 minutes, depending on the incubation time that we use. ELISA also incorporates 7 to 10 washing steps, while we have no washing steps. We just mix all the reagents and we let the device read it. Also, ELISA is semi-quantitative. In our case, it's very quantitative. We have an ELISA 2-log dynamic range, and in ONBI, we have about 4-log dynamic range. And also the analytical sensitivity is very high, limit of detection between 1 to 2 in ELISA, and in our case, it's about 0.04 nanogram per milliliter. So this is what we have, and this is the technology. And what we are looking for now is to incorporate this setup, which is very easy protocol and simple setup relatively, incorporated into a small footprint device capable of detecting very low concentration of biomarkers at point of care setting or low resource setting. So we are looking for collaborators in this field, strategic partners potentially, and I'm open to questions.
Thank you very much, Amos. This is exactly what this meeting is about. It's about finding the partner, finding the connections. I love the demonstrator. Well, it's not a demonstrator. I love your product. It's really great that you have already something that works like that. And like you compare with ELISA, tell us 3 main components that you think right now the industry, and we have here in the room people who make very stable lasers, great detectors, great integrated photonic circuits, and many of them providing surface activation processes. Please tell us a couple of challenges that you will be very happy going home with solved.
So as you can see, this setup, the device is a laboratory setting device. So currently we use free space optics to detect, to use a laser and a dichroic mirror and emission filters and a camera to use everything. I would very much like to use integrated optics and a VCSEL, for example, to use on top of the sample cell and integrate a fluorescent reader, a small footprint in fluorescence reader with this kind of device to make altogether a small footprint reader. That's something that we would like to have a challenge. The other thing is that I'm not sure if this is the audience to provide this help, but we have the device and disposable. So we don't have like the coffee inside the coffee machine. We have the coffee machine and we have the capsules, but we don't have the antibodies or the reagent. We don't have IP on them. So we are like an open access for this. We can work with anyone who have antibodies or would like to use antigens or have a specific DNA asset that you would like to use it to detect very low concentration. I didn't mention it, but for example, we are more sensitive than real-time PCR, about 4 to 9 times more sensitive than real-time PCR because we can detect fewer fluorescent molecules in solution than real-time PCR machine. So help in the optical setups and help in biological reagent, this is something that we are looking for.
Excellent. We have many people in the room that are going to be able to help you. Before doing that, there is another company that has similar challenges, so I want to give them the floor. So Amos, allow me to bring you to one of the most beautiful cities in this planet. I'm going there, I'm flying there tomorrow morning for a different business. I want to introduce you to the company Bioherent. Thank you very much for being with us today, Jonas. The floor and the attention of everyone comes to you. Tell us what's new about Bioherent, and let's afterwards compare it with Amos and see how the rest of the world can help us.
Yep. Sure. Can you hear me?
Loud and clear.
Okay, perfect. I'm going to share my screen. 1 second. Okay. Can you see the screen?
Crystal clear.
Okay, nice. Okay, so let me introduce myself. So yes, I'm Jonas. I'm the current director and the head of engineering here at Bioherent. Bioherent is a startup founded from the University of Malaga in 2021. We were founded by 3 different research groups, one clinical group, one biochemistry group, and one photonics group. And what we want to achieve, or what our vision is to develop integrated photonic biosensors for the in vitro diagnostic industry to help in the transformation to a decentralized diagnostic system. So, and our first mission is to do this for drug allergy diagnostics and offering an effective and accessible solution. So, what is the challenge or the issue with drug allergy diagnostics? So typically in the EU, around 120 people per year take antibiotics. And it's estimated that 10% of the population report a drug allergy. And the bad thing here is that typically 70 to 90% of those tests are misdiagnosed tests. You have a high false positive rate. And the bad thing about having or being reported or having a drug allergy is that you have to receive alternative drugs, which are more expensive, up to 100 times, and also are less effective for the patient, and therefore have a certain risk for the health or for the life of the patient. And that's why, or the higher false positive rate comes from the different drawbacks from the detection methods you have nowadays. So, for example, the in vivo methods, skin test or drug provocation test, are very risky for the patient, very uncomfortable. You have to monitor the patients for a long time and are therefore very expensive. And on the other side, you have the in vitro methods, which typically have a very poor accuracy. Some of them are not standardized and don't have a high accessibility for the patient. And that's where we hope to introduce our technology based on integrated photonic biosensing to offer a solution which de facto can offer a solution attractive for the healthcare system and also for the patient, offering a solution without risk and with a very fast diagnostics and correct diagnostic. So.
Thank you very much, Jonas, for being with us. Stay right there. We're coming back to you. In the meantime, allow me, well, many people are asking me if they can have also the chat. Yes, we'll share the chat with all of you. Don't worry. You don't have to worry about that. Let me bring to the table one person that is the first time at an Optica meeting, and he said that he can actually help MagBiosense with the free space optic-based interrogation of the chip. Gregoire Bonat, welcome to Optica. Please switch on your microphone and camera and tell us how you can support the work of MagBiosense.
Thank you, Jose. I'm Nick. Yeah, I hope it's all right. I just want to offer using the 2PP technology to print.
The beautiful thing of making these meetings live is that sometimes we lose the sound. Gregoire, tell us.
Yeah, sorry, I was speaking on mute, I guess.
Yes.
Sorry, I'm in a coffee. I hope it's okay. I was just offering to use 2-photon polymerization to print 3D print microstructures with a pretty high resolution, about 100 nanometer. And I don't know much about the use case here, but maybe it could be a good solution.
Amos, have you considered 2-photon polymerization, the job of companies like Nanoscribe or Vanguard or Heidelberg Instruments?
actually not, but I will be happy to explore it further.
Oh, you're going to get a lot of emails in your mailbox there. You are going to see. Thank you very much, Growar. Please keep coming to our online industry meetings. And allow me to keep bringing people to the table. Ravi, Amos, Jonas, let's now talk to the PIC community. Let's bring the PIC community to the stage and see how they can supply us. And before we bring the PIC community, one very important step is, of course, the surface activation of the PICs, because PICs by themselves are not biosensors. A company in the Netherlands called Surfix. Thank you very much, Luke Scheres, for joining the biosensing meeting. Tell us what you can do for others and what others can do for you.
Let me share my screen.
Always.
And I hope you can see my presentation now. Yeah, so
yes, we can.
Indeed, originally we were started off almost 14 years ago as a surface chemistry nano coating company. And in 2021, we made a pivot into developing a photonic diagnostic platform based on a small photonic chip, as shown on the slide. And we do this for point of care applications. And it has to do with the fact that currently, let's say 60–70% of medical decisions is based on lab testing. And most of the time, it's only one biomarker. It takes roughly 3 to 4 days before you get your outcome, and it costs roughly 30 to 40 euros per test. So if we go for point of care based on photonic chip technology, then we aim on lab level precision with multiple biomarkers at once. In this case, 5 result within 15 minutes, and an enormous cost price reduction compared to the current lab test standards. On the right, you see is very small chip. It's 3 by 3.5 millimeters nowadays, and we have a lot of these photonic spiral structures on it. It's a self-referencing design, and it's also a very long waveguide. So we have a lot of interaction with the sample, and we still use our surface chemistry. That's true. So we use it for advanced coatings to immobilize the antibodies. In this case, as you can see in the lower image on the right, to immobilize the antibodies in a very efficient and elegant way, solely to the waveguide structures. And that's one of the reasons why we are extremely sensitive. And in the middle, you see, for instance, a comparison of let's say, FDA or IVDR approved point of care products on the market, and where we are comparing the dynamic range and the overall sensitivity, and also multiplex. And you can see that we're really pushing it to the lower concentration ranges into the femtogram per ml range. And this is not just a story, as seen in the orange, can be seen in the orange bar. We just recently successfully were validated by some top-tier diagnostic players in blind studies. And so we are doing very well, and we're very happy with the progress that we made.
And we are equally happy for having you in the room, and thank you and congratulations on that pivoting to provide the entire solution, not just the surface activation, to your customers. This is great, Luke. Jonas, there is one corporate member, Wuchan Household, who has something that would like to comment to you and see how you can work together. Stuart Marfarkar, a really good friend of Optica. Tell us what's on your mind.
Hi, thank you, Jose. Yeah, I was just curious, Jonas, about you were saying that obviously integrated photonics has a lot of capability for improving access to diagnostic capabilities, but you were talking as well about improving the results of the allergy diagnosis and reaction diagnosis to that. I'm just curious about the methods that you're able to use to improve that relative to traditional allergy diagnostic results. Yeah,
sure. 2 ways. I mean, you have to tackle this question with the surface chemistry, of course, because that's a very, very important part, but also let's say the measurement environment. Okay, surface chemistry is very important if you want to have a very sensitive and a very specific detection. So having a specialized surface which can improve or which has an advantage against the other methods used in the market is very important. If you can offer a surface chemistry which can be more sensitive and more specific than the other techniques, That's a competitive advantage. So you have the potential to go to the detection limits which are relevant to make a better detection. But then also, of course, you have to consider the instrument or the cartridge side. You have to offer a solution which fits more or less the market needs. And I'm talking about, okay, what is typically the context if you want to have a drug allergy detection, or if you want to verify that someone's allergic to a certain drug. So typically the doctor is asking, okay, I want to give you this antibiotics, but I don't know if I can give it to you. So I'm going to make a test. This test is going to take some days, and that's very, let's say, not optimal for the patient, because he has to wait until he gets the test results. So he's denied the initial antibiotic phase. Let's say it this way: if you have, for example, a device which you have in your write a design for, let's say in the future, in the next, say, a point of care device where you can make the test and directly say to the patient, okay, here, take the antibiotics and you can go home. That's a competitive advantage against the technologies you have in the market, to have a result which is precise within some minutes.
I see. So you're looking at systems where you can directly detect the immediate emergence of the relevant antibodies that you would expect a person's body to create in reaction or in response to that specific potential allergen.
Correct.
Yes.
Yes.
Correct.
Okay, that's really interesting. Thank you for that.
Thank you, but Stuart, you really have to follow up with each other. Let's continue. I want to go to a beautiful city, Jonas, because I want to talk to Luke a little bit more. Luke, do you know city Milan? Milan is a beautiful city. I love it. And I want to give the floor to Stefano Prandoni from FiberTech, who has a question for Luke in Surfix.
Thank you very much, Jose. Hi, Luke. I have a question about your device. So I think you used a laser inside the device, right? And if so, if you can tell us more about the laser you use, if you can.
Yeah, so we use a Vexel. That's also a laser, right?
Yes,
Yeah,
but
sure.
I covered the surface meeting one.
And we tune its wavelength over 3 nanometers, and that's we use for the Saxena for readout. And I only had 2 minutes. I forgot to mention that we got the chips from Leuermann. So Arnoud, I noted you. Is this answering your question, Stefano?
Yeah, sure. If you don't mind, maybe I will get in touch later. I'd like to discuss.
Believe me, that was exactly the answer that Stefano was expecting. I think, Stefano, could you tell us why you think big cells are the right sources for biosensing?
Is that a question for me or for Stefano?
It's for Stefano Prandoni.
Oh, okay, sorry.
So actually, there are different reasons why you can use VCSELs for this kind of tests. We don't know all the details because the projects we have with our customers are all covered by secrets on customer side. What I think is most of the customers using for biosensing the lasers use the swept tunable because they can hit. On one side, they can hit different wavelengths, so they can test different also materials or substances. And on the other side, I think there's some kind of technique to use the sweeping, so the modulation, to subtract a background noise. And this makes the device more sensitive.
Amos, Ravi, if you are looking for a person to guide you on how to use VCSEL to do the right interrogation of a biosensor, it doesn't get any better than Stefano Prandoni. I hope you have a good discussion afterwards. And Luke,
Thank you very much,
question for you. One question for you and almost for everyone. Why silicon nitride?
Why silicon nitride? Because it's very nice optically transparent in the wavelength region that we would like to measure. And the biomolecules do not have a lot of absorption in that region.
We have in the room, and they are going to give us a short update, two of the key foundries on silicon nitride technologies, LIGENTEC and Lionics. I'm going to give them the floor for 1 minute presentation to each, and then we're going to have discussion what needs to happen on silicon nitride to satisfy the need of the people in the room. Let's start with LIGENTEC. Anton, Stroganov, great to see you today. I love your beard. You look fantastic. Tell us about silicon nitride platform for LIGENTEC.
Okay, sure. Nice to be here, and let me just set up the screen. I hope you see the slide now.
Gorilla Glass clear.
Okay, that's perfect. So let's start first with a short reminder on what we do. So our main business and expertise is manufacturing of photonic chips using silicon nitride as a main material, which is known for its broad transparency window and, of course, for very low optical losses. So we supply photonic integrated circuits for your applications, including sensing, microscopy, and spectroscopy. And generally, to realize performant optical sensor devices, we also developed special sensing modules with local waveguide exposures of different kinds, which allow for highly sensitive chips for various applications, including label-free biosensing, Raman spectroscopy, or even bioimaging applications. We do manufacturing at any quantity, from early prototypes to volume production, while keeping the state-of-the-art performance and cost, which allow viable manufacturing of even single-use devices, which were discussed before. And generally, to make your life easier, we also offer a broad range of components, including mature process design kits with pre-developed building blocks. And besides, we bring an opportunity now for integration with other materials. So our platform has been used, for example, for realization of compact external cavity lasers with narrow alignments at very different wavelengths, spanning from visible to 2 and 3 microns. Sorry. We also now provide direct integration of photodiodes onto your chips using microtransfer printing, as well as hybrid silicon nitride and lithium niobate devices. And generally, all of these features, they open great perspective for advanced performance and for complex functional devices for sensing and other applications.
Thank you very much, Anton. Just before your presentation, in the beginning of the presentation, Ravi from PacBio commented about how it would be improving to add modulation technology to their technology. You said, and also Photonics West, you were showing your thin-film lithium niobate modulators on silicon nitride. Could you tell us a bit about what is the current wavelength range in which they operate, and at what wavelengths can you provide modulation on chip?
Yeah, that's a great question, and especially in the context of the hybrid devices, because we all can imagine that if it's pure lithium niobate or pure material of just one kind, it's much easier because you typically just in one plane. But if we speak about hybrid devices, it's good because you can really have much more sophisticated devices with the best performance of the different material platforms, but at the same time, it brings the complexity of combining materials and making sure that you have waveguides which allow you to really function and have an optical interaction between different layers. So here, answering your question, we obviously start focusing on the telecom wavelength ranges. So C-band and O-band are already fully addressed, and we offer it as a part of our standard offering. And the next step for us is now going a bit shorter in the wavelength range, so to 900 nanometers, and below that, all the way down to 600 nanometers.
We are going to comment on that in a bit, but afterwards we have LIGENTEC. Let's also hear from Lionics, a big partner of many of the biosensing companies. It is great to have Arnoud with us from Lionics. Arnoud, tell us your side of the story in one slide. What can you provide, and then we'll follow up on what needs to happen to silicon nitride in biosensing.
Yes, thank you all for the invitation.
I cannot see your slide. It just came and disappeared.
I was sharing my slides. It's now sharing very well. So I'm Arnoud Everhard from Lionics International, and we indeed do silicon nitride integrated photonics. And here we do it for your biosensing business. In Lionics, we have the full vertical integration where we start from a design. You can start it in chip design, just optical design on the chip, but you can also do complete full system designs for the complex systems. Then we bring it to fabrication, something we do in-house for our silicon nitride foundry. And we do this from small to medium volume production facilities. The next one we can do is to do the assembly, because you don't need the chip, we just need the light in the chip. So we can couple optical fibers, lasers, detectors, modulators, and all these kind of electrical and optical components. And as the last, you don't only just need the connections, but you also need the full packaging, the mechanical, electrical packaging, and so we also do the electronics for a full turnkey solution in the end. Because what we are focusing at LIGENTEC is the silicon nitride waveguides, which we call triplex at LIGENTEC. And what advantage is there is the ultra low insertion loss and the low propagation loss. So we can actually have your light going into your chip, processed on your chip, and get out with plenty of light left in the end. We have a mature and stable process at LIGENTEC because we have 20 more years of experience and we have already plenty of showcases. We have an interdisciplinary samples and we do the full circuit integration because we can do all the parts that you require. And the silicon nitride advantage is we can go down to the visible light, not only infrared like everyone else, but also even down to 400 nanometers in the blue region. That's the advantage of silicon nitride can offer for this biosensing. So we have several examples of things we did in the past. First of all is the evanescent field sensor from Surfix, that Surfix is actually using, that they showcased 2 presentations back. Second is the OCT, optical coherence tomography, where in the past we did spectral domain OCT, but now we're actually pushing towards a swept source laser to make it a really attractive commercial viability to do OCT on a small scale in a handheld way. What you can not only do is have the optics, but also introduce and integrate the microfluidics. So this is one where we use a flow cytometry to have the light coupled into the chip, but also the fluidics interacting with the light. And last, there's a lot of other optical systems and functionalities that you can do. You can build your own module, you can design your own, you define your own module. So anything with light we can do, for example, this spectrometer on chip. So if you have any questions, we'll be free to chat.
Lots of questions, lots of suggestions we have now in the room. LIGENTEC, Lionix, Surfix, and 2 of the integrators and success stories of silicon nitride technology. So let's have a quick discussion with all of you. And to kick off the discussion, let me go first to CSEM. Andreas Volker, all the way from beautiful Switzerland, even more beautiful Neuchâtel. Tell us what's on your mind.
Yes, I think we had hello everybody, and thanks for all the great contributions today. Listening to the question before that we had from Milan about sweeping the vexel or the wavelength, because this will basically allow you to hit different targets. On the other side, you could look at the detection on the PIC. And here I was wondering and looking at also what LIGENTEC showed us, the combination with a thin film lithium niobate. Here you could also envision that you quickly tune the spectral response or the spectral filter, what you're detecting. And I wanted to know if this is something that Surfix is investigating.
Look, instead of tuning the laser, just have a broadband laser and have a tunable filter.
Sorry, I missed the question a bit.
Instead of tuning the laser, what Andreas would like to propose is that you use a broadband laser and then you have a filter which you tune. Is that something possible? I think, Andreas, the challenge here will be how narrow the filter can be. Do you have some numbers on that? If not, we could go to Delta Optical thin film, who's in the room, but I think it's also another solution. But before we do that, there is another question in the room. The question is coming from Guchan Hausgo. Stuart, tell us, what do you have in mind?
Hi there. So obviously, G&H are more associated with the production end, particularly with our sort of contact with integrated photonics so far. And a lot of the applications that we have in mind for this sort of technology are extremely sensitive to system losses and similar. So I know I've seen on a previous LIGENTEC presentation, I'm sure Lionix and other foundries can also offer very similar kind of figures of fiber to chip coupling losses of extremely low numbers, so less than 1 dB per facet. Most of the experience I've personally had with trying to do this sort of work in experimental situations has been extremely sensitive to misalignment and has had sort of a much higher level of loss relative, much higher than 1 dB. So I was wondering really whether this kind of thing is how easily achievable this has been on purely chip to fiber, or is this mostly being done with because I've also seen interposers from Fix and similar organizations that do spot size conversion. Sort of wondering what the overall approach is to this.
I would like, I think both Anton and, well, Anton, you go ahead.
Yeah, sure. So it's also a very good question, and coupling light from the outside world to the chip was always a bit of a struggle for the whole community. But answering your question, so basically how we at least approach it, that we do a special spot size converters, which are done in the process of manufacturing, so on a wafer scale, and therefore allow you to expand the mode field diameter directly on a chip to match the mode field of the standard single mode fibers, SMF28 and C-band, or just other single mode fibers. So we do it both for the telecom wavelength ranges, and recently we also introduced a component which allow you to do the same for wavelength range of roughly 800 nanometers to 900 nanometers, and the performance indeed could be well below 1 dB per facet. So it can be both. It can be fiber to chip, or it can be introduced or manufactured also to be used as an interposer between a fiber and a different type of a chip.
Thank you very much, Anton. If anybody else would like to comment. If not, I would like to keep bringing people to the room for discussion. And with that, I would like to go to TNO. TNO is the R&D Center for Industrial Research and Scientific Research. And they have done for many years lots of work on biosensing at the TNO Halls Center. Thank you very much, Natallia, for being with us today. Tell us what you can do. Most important, tell us how the others in the room can help you. The floor is yours.
Wonderful. Thank you very much, Jose, Elena, for hosting this meeting. So indeed, today I would like to highlight a specific role that TNO plays in integrated photonics for biosensing. And I would like to bring to a discussion that we have in the area of early detection of biological threats. So as you know, of course, today, and therefore we are in this meeting, biosensing is mostly confined to centralized lab, and this is really far from where the results are needed. And therefore applications such as healthcare, pharmaceutical industry, but also defense would really benefit from having real-time, multiplexed, sensitive, but also miniaturized and low-cost biosensing. And that holds for many applications in chronic infectious disease management, drug development, personalized medicine, as well as in a very specific area of detection of biological threats and active countermeasures to that. And this is really where integrated photonics bears this very lucrative promise to revolutionize the area of biosensing. And therefore, we are all here in this webinar. But let me describe a little bit more the strategic role and very wide, versatile role that TNO has been playing in the area of biosensing in a wider sense. We are known for over more than 10 years of innovation for medical device companies, and via the partnerships and open innovation form of working, we assisted many of the material development companies that are delivering materials for the medtech, as well as we accelerated device innovation for medical device companies like Philips and Bambi Medical and more. And these applications range from simple vital signs and wearable ultrasound to electrical and photonic-based biosensing. And again, this is the topic of today. So let me highlight one company which is known for you, to many of you, which is Delta Life Science. And Delta Life Science is a spin-off of TNO from 2018. Delta Life Science focuses on label-free evanescent field sensing, and it's backed up by over 15 years of R&D work at TNO and beyond of it, a strong patent portfolio in expanding it. And its target market, life sciences, the company is financially stable and they secured Series A investments. And in the heart of this technology is asymmetric Mach-Zehnder interferometer, which allows for label-free measurements of 16 analytes in a sample. Delta Life Science can handle both pure and complex matrices, and it can handle volumes down to 25 microliters. It's focusing mostly on kinetic studies, but it can also do quantitative measurements. And Delta Life Science has this very strong and down-to-earth promise, be faster and accelerate research by 4 times, by delivering more data from the same sample, by delivering faster data, and by accelerating your assay development. So now, you know as well that biosensing is a very complex and multidisciplinary topic where sample handling, biofunctionalization, detection, as well as integration of light source and on-chip integration. These are all very complex disciplines requiring significant investments. And therefore, we are working together with the partners in large-scale programs and ecosystems. For example, in NextGen High-Tech nationwide program, we're working with many partners on developing generic fluidics module. In Photon Delta National Growth Funds, there are many companies that are developing different types of integrated photonic solutions for biosensing. Biofunctionalization, we just started a separate project at TNO using dry and scalable technology, but also within the European project, Photon Med, there are partners working on biofunctionalization and integration, so on-chip integration of light source and detector. We are tackling at PITC, Photonics Integration Technology Center in Eindhoven. So really, the Dutch facilities feature really the suite of different options that include silicon nitride fabrication lab at Mesa Plus Institute in Twente, indium phosphate lab at Eindhoven University Technology, and chip integration is happening at PITC, Photonics Integration Technology Center, and CITC, all in the Netherlands. And of course, we are embedded, very well embedded in manufacturing ecosystem. The representatives are here in this meeting, Lionics, Smart Photonics, and FIX. And we are very happy that the fabs fabrication facilities are scaling up. That is really important. There is a new origin, pure play foundry in Enschede for silicon nitride, which is extremely important because it offers a low loss waveguide material. And recently, there has been injection of money via CHIPS GU for a pure play indium phosphate foundry that will be located in Eindhoven. So there is an ecosystem and there are many interesting companies in this field. Let me come back to the application that I would like to highlight today in this meeting. That's a real-time biosensing for defense. Early detection of so-called weapons of mass destruction, which includes low volatility chemical agents like nerve agents, novichoks, high potency opioids like fentanyl, as well as high priority pathogens. Think about anthrax. So biosensing is needed for surveillance, for example, in air and in water, as well as for pre-symptomatic detection in different human matrices, different body fluids. And earlier, TNO already proved one of the concepts for detection of exposure to nerve agents, for example, organophosphate. Butyryl cholinesterase is one of the high potent biomarkers of exposure to such organophosphate, which eventually lead to neurotoxicity and paralysis. And TNO leveraged its knowledge on optical asymmetric Mach-Zehnder ring resonator, and TNO demonstrated detection of butyryl cholinesterase at clinically relevant levels within the minutes. And a current use case that we are elaborating together with our partners, that's detection of high priority pathogens, such as Bacillus anthracis, where fast detection is needed in air and in water. The device needs to fulfill WHO criteria, so it needs to be affordable, it needs to be portable, it needs to be fieldable, rapid, robust. And Tino is leveraging its expertise in integrated photonics and on-chip integration for that particular use case. So really, to summarize here, we are all here together because we do understand that there is urgent need and sensitive, multiplexed, real-time, but also low-cost biosensing for healthcare, but as well for defense. And integrated photonics, it has this lucrative promise to revolutionize the area of biosensing. And there are really very promising startups, as we have seen today, in the Netherlands and in European arena. And earlier, Tino already proved the principle of detection for chemical agents. And currently, we are elaborating the use case for detection of high priority pathogens in air and water. That case is very interesting because actually you can start from a relatively simple matrix, air and water, and later during the product development, you can roll out that to the healthcare applications, which are obviously much more complex and require much more strict regulatory clearance. So an overall, a Dutch high-tech and integrated photonics ecosystem can really enable technology scaling up. So as we all know, partnerships drive progress, and we are really open for collaboration. So please get in contact with us if this particular use case triggers your interest.
Thank you so much, Natallia. This is actually very good, and this is a different way to see all the biosensing, especially for the Canaries. I'm very happy that you bring it here. So one of the first questions is like, what is the advantage of having these different architectures? You know, a Canary versus maybe a plasma surface or a different, you know.
Well, I think it's a very good question. So from our perspective, as I mentioned in the talk, so we already have over 15 years of experience using ring resonator structure, and that basically the optics is already very mature. That's really high TRL level, and the chip that I show in my slides also has been manufactured by Leuermann. So I think if you would really like to have a faster way to market, you would choose this kind of structure that can be designed very nicely, that has been proven already, can be manufactured. So I don't want to make anybody uncomfortable, but I do believe that plasmonic structures are a little bit farther down the road. So perhaps for the future, that can offer an advantage, such as being perhaps more sensitive, though it's not really, I think it's not really black and white. And a lot of things depend on the chip design. A lot of things depends on the sampler preparation, and a lot of things depends on how you do biofunctionalization. And we have Surfix here, so they know. So, and I think it's overall system requirements. You can split between different functions. Partially, you can address your sensitivity and specificity by sample prep. Part of it you will address via biofunctionalization, and partially you can address it using better design or different type of design of photonic structure. So everything has pros and cons, and it's up to the architect to make choices.
Yeah, this is one of the things is because defense is based more in plasmonic. Most of the biosensors are developed, and this is something that is good to see. There is also like, have to be ultra rapid, have to be on site, a detection on airborne, detection on waterborne. So it's all complicated. But so Ravi, actually, if he wants to talk to you, he got some questions for you. So Ravi, could you please open your camera and your mic?
Yes, thank you. Thank you for that lovely talk, Natallia. I was curious, I think you mentioned earlier in the chat, I was browsing through that you had some ideas of dry surface functionalization. Could you just comment a little bit more on that and how you were leveraging that in your technologies? Thank you.
Yeah. So absolutely. So we started, I mentioned, of course, large national programs where Tino is participating together with our partners like Leuermann, like Surfix. Dry biofunctionalization, we started internal Tino activity to explore using cold plasma technology in order to immobilize, in the first instance, the linker layer. And then perhaps in the future, we can directly immobilize aptamers or any other biorecognition molecules on the surface.
Excellent. So
Thank
yeah,
you.
so Yeah. So this is one of the things. So is this like is it possible a collaboration in between what you are doing and Ravi? Is it some way to to overlap somehow, do some extension, is it?
I think I'll connect with Natallia offline.
Yeah, I think that would be the best.
That's what I
Yeah. So
wanted
now
to hear.
we saw each other. Yeah, of course. Yes, absolutely. Happy to hear. Yeah, happy to connect, Ravi. Yes. Yeah.
Yeah, excellent. So Anton also has some comments to make. So Anton, can you please open your mic? Anton LIGENTEC? Yeah.
Yeah, I'm not sure whether it was a particular comment. I was rather following up a bit on the presentation, stating that yeah, we are as a company very happy to support any peak development, or if any company really wants to go from their free space-based solutions or fiber-based solutions into integrated photonics, because I see that it's quite a topic even during today's meeting. We will be more than happy to support this journey.
Wonderful. Thank you so much, Anton. Yes. I believe we're together in PhotonMed project.
Yeah.
Yeah.
Yeah, absolutely. So I would like to take actually the opportunity actually to come to bring to the room now Tom Campbell for Airware Incorporation. So he's this company is focused on real-time biochemical monitoring, and I think this is the right moment. So Tom, can you please open your mic, share your slide, and tell us about Airwave?
So thank you to Optica and for inviting us. And I had a chance to review our technology with Jose at the Photonics West show, and hopefully I can channel some of his enthusiasm today. So we're making blood sugar awareness as common as your heart rate and as easy as pulse ox. And what we have is 13 issued patents that describe our unique approach to sensing analytes and liquids. Our focus is glucose. And we're looking at it for interstitial fluid in skin and accessing it by transmissive sensing to delivering a solution to the stack challenges in non-invasive and in vivo measurement. So the biggest challenge is overcoming scattering noise. The second is avoiding water absorption. The third is using a constant reference. We're tracking interfering analytes with an additional laser. And all this with transmissive sensing delivers repeatable and accurate glucose monitoring. So with a 50-year background in gas sensing, when we approached this, we recognized we had to solve the scattering noise. So we clamped to a 3 mm fixed height, like a pulse oximeter on the finger, but we're pinching at the hand web for a controlled interrogation volume. So what we're really doing is we're employing our narrowband alternate and sequentially pulsed lasers for transmission through 3 mm of skin. And we use this to achieve high signal to noise through differential spectroscopy. So our target lasers are centered on second order overtone absorption peaks to avoid water absorption. And each laser follows the exact same path that allows us to cancel out common Rayleigh mean and geometric scattering noise. And this allows us to lower the noise floor. So the heart of our system is the electro-optical engine that many people have alluded to. And our key is we're using very narrowband quantum dot DFB lasers mounted on a waveguide combiner that delivers our highly controlled single path emission. And that small laser engine then becomes a small component of our handheld clip. We anticipate a clinical plan that places our portable units with a broad cohort for a week-long evaluation. And from data from that broad clinical trial will allow us to finalize our front-end ASIC. And the ASIC driver enables a downsize to our handheld clip. We'll be doing an FDA submission under an ISO spec for blood sugar monitors. And there is an existing reimbursement code for both optical blood sugar sensors. It's been sitting there since 2002 and no one's delivered a product to date. So we're really looking to provide no pain, no blood, and a simple blood sugar measurement. Thank you.
Thank you so much. So if we have any question for Tom. So Tom, this is one of the things that is happening. So what makes you different measuring the glucose compared to everybody else?
Well, this comes back to 1,000 efforts have been made to tease glucose signatures out of the skin. So you have to get through layers of skin down into the interstitial fluid. And the biggest issue is where most of the technology is focused on first order absorption overtones. They're highly overshadowed by water. So you're essentially measuring water and not the analyte in the liquid. So this is where it comes down to this stacked set of challenges. If you don't solve them all simultaneously, you'll never get to repeatable and accurate measurements. So our whole basis is trying to work within that stacked solution. And our breakthrough really is this alternate and sequential pulsing that allows us to, we ratio the peak values for a pulse pair. And when we ratio those with differential spectroscopy, we cancel out the common Rayleigh mean geometric noise. So that's the real breakthrough. The rest is a kind of a long slog through how we get that very specific narrowband light pulsed into a very, very small form factor. And what's really come about is that this last Photonics West show is I've heard more yeses to what we're looking for than ever before. And there's been so much done with silicon nitride waveguide work that I think it'll be a simple matter. The real question comes down to whether we have to commit our laser to a hermetic package or whether we can directly mount the die onto the waveguide and save that packaging step, as long as we can guarantee relatively tight plus or minus 1 nanometer peak wavelength out of the lasers.
Yeah. So I'm pretty wonder here, like who can help you with that. Thank you so much. And our final keynote from today is David Duffy from Quanterix. So Quanterix is really revolutionizing the biomarker detection with ultra-sensitive digital immunoassay technology. So the floor and the attention is all yours, David. Thank you so much.
Thank you, Elena. It's an honor to be participating in this group. And so I'm going to make the case for digital detection of proteins as a way of biosensing that can revolutionize things. And when I say digital, I mean basically counting single molecules of proteins. So this may be preaching to the choir a little bit, but why digital detection? It comes down to 3 things: sensitivity, and sensitivity. And kind of when I look at molecular diagnostics in the last 70, 80 years, it's really been driven by sensitivity. And Steven Pinker, a hero of mine, he agrees. So if you look at PCR, we've heard about next-gen sequencing, CAT scans being the more sensitive kind of X-ray, MRIs, and thing that we work on, which is sandwich immunoassays. They've all the innovations have been driven by making the technology more sensitive. And the hypothesis behind Quanterix was if you can count single protein molecules, then you can't really get a more sensitive immunoassay. So I always have an iceberg slide in my presentation, so I'm just going to stick with that. And back in 2007, really measuring proteins was the tip of the iceberg. There were about 190 FDA-approved protein tests out of what we knew was like 30,000 genes. So we weren't detecting everything, and the water levels are kind of picomolar concentrations, which is about 15 million proteins in a finger stick of blood. And with digital detection and Simoa, we've been able to kind of drop the water level by somewhere between 5 and 6 logs, and starting to measure things like neurological markers that previously could only be detected in CSF, in blood, and prostate-specific antigen in men who'd had their prostate removed. So I think kind of the principle has been proven in terms of the sensitivity improvement you can get by doing digital detection. So what's this given us is really been able to change the paradigm of healthcare in that when you look at kind of two axes, you've got the status of disease of someone and then how you detect what sample that you get. And, you know, unfortunately, we've really been focused on late stages of disease where the biomarker concentrations are high and samples that are quite hard to get hold of, either, you know, biopsy, a spinal tap, or having to use imaging. So by making things more sensitive and counting single proteins, you can kind of move things down to the bottom left where you're detecting these markers of disease, you know, when people are healthy or if they're just starting out and pre-symptomatic, and in samples that are easy to get hold of. So the big story behind Quanterix has been using this capability to kind of detect neurological conditions before symptoms arrive with a blood test, which, you know, didn't exist before we were doing this. And just to kind of give one more kind of slide on the motivation, and this struck home with me last year, in that if you went on Twitter, there were people who were basically begging the FDA to approve a drug for ALS, and because they were seeing their friends and family benefit from this drug in clinical trials. And in April of last year, I think it was 2 years ago now, actually, the FDA approved this drug purely based on the measurement of a biomarker of ALS that we could measure in blood using Simoa. So no clinical outcomes, just based on biomarkers. So for everybody on this call, if you can develop technologies that allow people to measure molecule sensitivity, you can have a direct impact on people's lives. So that's been nice. So what do we actually do? So it's a pretty simple but powerful concept. So we start in the top left here. We start with beads, little beads. They're about 3 microns wide, coated in antibodies, and we mix them with a sample. And kind of thermodynamically, they literally just suck every protein out of solution. That's kind of the clever thing. And then we form a kind of classical sandwich where we have a second antibody that's got some kind of label on it. And when you're at a very low concentration, you actually have fewer protein molecules in the beads. So they distribute in Poisson style, and you get a bunch of beads with nothing on them, and you get statistically pretty predictable numbers of molecules, beads with 1 molecule or 2. So if you can now detect a single molecule on that bead, you'll suddenly kind of drop the water level, those 3 logs. And so the kind of challenge as a technologist, and you know, we've worked very closely with people on this call and others in the optical industry, is to kind of interrogate those beads one at a time and determine if they have a molecule on them or not. So we spent most of our time doing imaging. So this is more kind of classical multi-channel fluorescence microscopy. We've heard some about that and using microfabricated microwell arrays to do this. But more recently, groups have been using flow cytometry type technologies where you're kind of imaging the beads one at a time, but using lasers and slightly more expensive detectors. But what we end out of that is kind of the fraction of beads that are on or off, and we convert that into the average number of molecules per bead, which is effectively a concentration. So the kind of the tricks behind making this work was we were normalizing the number of molecules to the number of beads. That helped precision. We had very highly efficient capture with the beads, as I mentioned. And we use chemistry to kind of do this signal-to-noise ratio work where we're generating thousands of fluorophores for every binding event. And that enabled us to move to much lower cost illumination and detectors, which I'll come back to. And it's all based on kind of the classical immunoassay that was invented kind of back in 1971. So we could use all of the reagents that existed to do this. And as I mentioned, you know, our technology is called Simoa, and we do this using microwell arrays. So where's this all going? So, you know, the challenge kind of us in the future is right now, these instruments are great for centralized clinical lab testing. So the instruments are big, and they usually require people in white lab coats to run them. But where we want to get to in the future is a much simpler, lower cost, more kind of democratized technology. So kind of benchtop diagnostic systems, kind of what we would call point of care, kind of much simpler, higher throughput research instruments. We've heard already just in the last presentation about wearables, and there are wearables for measuring blood glucose biomarker in blood. They exist today, so why not single molecule levels? And we all saw the impact during COVID of kind of home testing. So that's kind of the vision and the technologies that we've been trying to develop. So I've just taken the last few seconds to say, we've been working on this. We've been trying to solve some of these problems. There are plenty of problems to solve here. One we described at Microtas in Montreal last October, where we're using digital microfluidics to handle very large sample volumes, 100 microliters, but then do the manipulations on much lower volumes and kind of shrink that instrument down to a microchip. And probably most relevant to this group is imaging. So we really need low cost, miniaturized, simple, manufacturable imaging. And that could be either flow or it could be kind of microscopes. And so we've been doing work on trying to kind of strip everything out and using LEDs, plastic optics, to be able to detect these beads and count single molecules in a multiplex format. So we described some of this work at SPIE now a couple of years ago. So if you'd have asked me 10 years ago, could this stuff be done? I would have said, no, you're crazy. But the technologies that are coming along on miniaturization and optics and sensitive detection, they're getting there, where I think some of these kind of more democratized technologies will see the light of day. So hopefully I've stuck to my 6 minutes, but happy to take questions, and just should acknowledge that the point of care work has been funded by the NIH, and we have collaborators at Boston Children's and in Toronto.
David, time runs, but time flies when you're having fun, and you gave a fantastic presentation. We definitely saved one of the best for the last. It was a great presentation. What we want to know from you, clearly, is that you're seeing technology being developed in a way that you're making something unthinkable possible. What would you expect from the people in the room? What kind of challenges would you have for us?
Yeah, when I think about it, I think I kind of try to outline them there at the end. I think there is this ubiquitous need for kind of multi-channel fluorescence, ultra-sensitive detection technologies, and it can be imaging, it can be flow. I've kind of heard examples of both on this call. So, some of the, the high-end flow cytometers, they're incredibly well suited to this in terms of the number of channels, the sensitivity, but they're big and expensive. I think the expense side is what is really holding things back, and the size and expense tends to go together. So I think, as if people can, as we've heard today, integrate light sources, optics, all onto chips to make things in very small packages that are cheap, that could then be scaled, because your numbers go up orders of magnitude when you try to go mainstream into diagnostics. I think that's where this community can really help. And as I said, I'm seeing more and more of it. So, yeah.
Thank you very much, David. Please stay with us, please, all of us. If you want to, please stay with us. I need to close the meeting because we're already 7 minutes out of time. We did our best to keep it in time. But for those of you who want to stay for a quick after beer or after non-alcoholic beer discussion, please stay on. For those of you who join and follow the meeting both in Zoom and YouTube, thank you. Thank you so much.